we're going to move on to our third section on diseases, communicable diseases, and others not. My first guest is Philippe Froguel. Please come up, Dr. Froguel. He is professor at University of Lille 2 and Imperial College in London. He is an MD, researcher in endocrinology. In fact, he's a world leader in research on the genomics of diabetes. He has created two very important research units, one at the CNRS Lille 2 Institut de Pasteur Lille, and the other one, the Center for Genomics and Proteomics at Hammersmith Hospital Imperial College. The 4 Ps, diabetes is a complex disease. How can we make it more personal and the treatment? 7 minutes. Thank you, doctor.
Thank you. As you know, there are 300 million people with diabetes these days, and this number will double in the next 30 years. When I was a resident some time ago, we used to weigh the patient. And when the patient was obese, we used to give them a drug named metformin. And when the patient was non-obese, was lean, we used to give them another drug, the oldest drug, oral drug for diabetes, named sulfamide. Today, we still weigh the patient, but we give all the patient the same drug, metformin. And when it fails, as it usually does, there's no clear guideline what to do. And as a consequence, diabetes worsens. A large proportion of patients develop complications, and they still die early. So from this very low point, is it possible to develop a more personalized diabetic medicine? And my response is yes, because we have some proof of concept that it may work. 10 years ago, my group and another group in the UK identified 2 genes responsible for very early onset diabetes. It was a gene encoding for potassium channel of the insulin-secreting cells. Why it's important? Because it changed everything for this patient. This patient had insulin from the first days or months of their life. It has been possible to stop insulin, to give them sulfamide, and of course it changed their life. What did we learn in the last 10 years? First, that these patients, when they received sulfamide, had normal glucose values, and they still have normal glucose values. So they are virtually cured from diabetes. It's good for them, it's good for the society, we save a lot of money. Secondly, they are more numerous compared to what we expected at that time, and many patients, even with what we call garden type 2 diabetes, coming from diabetes, have this mutation, and when we recognize these people, we give them sulfamide, we have exactly the same result, virtual cure of type 2 diabetes. So, is it possible to do better? Yes. First of all, we believe that about 5% of patients with diabetes have this kind of genetic diabetes, and they can be recognized if we want to do it. Secondly, we believe also that it is possible beyond to treat better a large proportion of patients with diabetes if we are able to recognize their specific disease. So to do it, we need to meet different objectives. The first one is to be able to diagnose this patient, to understand why they are diabetic, what is the etiology. The second, we have to elucidate the physiology of their own disease, not the disease of the other guys, but this one. And third, we have to be courageous and to give them a specific treatment, not the treatment of everyone with diabetes. So for the recognition, the diagnosis, some groups, like my group, have developed some approaches, genetic approach for the diagnostic. And we developed targeted resequencing with NGS approach, very inexpensive, very quick, 3 days, and also extremely sensitive. Secondly, when we don't know exactly what is a gene, and it's a new gene, we developed very inexpensive, very sensitive whole exome sequencing approach, about 400 euros to do the work. And by the way, Madame the President of INCA, it's easy to translate that for cancer. And I have to tell you that if I had a cancer, or if my mother had a cancer, I wouldn't understand why we have to wait for 2019 to generalize this thing. My group in Lille, my equipace, we can do that for you if you want. And we can do that today and not in 2019. As a patient, why we have to wait for something that is available in France? Of course, maybe not in one of your 28 centers, but in some centers paid by the government through the PER. So first thing, we have to recognize it, and we can do it today, not tomorrow. The second thing, we have to understand why these people are diabetic. And we have developed some integrated approach with metabonomics associated with whole exome sequencing to try to understand when you find a mutation, because you always find mutation, what exactly is going wrong in this patient. And secondly, we developed, we are not the only group to do that, we developed stem cells, somatic stem cells, to test drugs to the cells of the patient. So the insulin-secreting cells of the patient, the hepatocyte, the muscle cells of the patient, from the blood, we develop multipotent stem cells and then we derive. And that is a kind of testing drug for every patient. Of course, it won't be possible for every patient, but for some patients it will be possible, and it's a way to try to have new treatment. And of course, as I said before, we have to test it and to try to give this patient a new drug. So is it possible to move to personalized medicine diabetes? Yes, we can, but we have to just to do it.
Thank you very much, Dr. Froguel. Would you like to answer to the attack here?
Yes, I will answer. First of all, the idea is not to perform sequencing on the 28 platforms. And the question is not sequencing, of course, it's the analysis, the bioinformatics, and the algorithm. So you need a knowledge of cancer and cancer results with the treatments to implement the system. So I have 10 offers of labs who want to collaborate to perform sequencing. My question is, who is performing the best analysis and performing algorithm that can be useful to the patient? And as we need big data, we need to organize it for the world territory. I think it is not a center approach, it's a national approach if we want to be efficient.
Okay.
So, but I will look at your offer, Dr. Froguel.
In
Yes.
You know, in genomics, there is no specific disease. There's the genome and the abnormalities of the genome, and especially for cancer, somatic genome. We are working on ovarian cancer, and we have colleagues from Montpellier, and they are desperate because they think they are going too slow, and they are thinking about creating companies to do the job and to move to personalized medicine. I'm totally against the bureaucratic view of that. In favor of some example that works and can be generalized later, and not the opposite, button up and not the opposite. And I think my colleagues and I and others, we will prove that we can do well.
Okay, well, maybe at lunchtime where you'll have a chance to pursue this discussion later on. Yes, you have a question? Yes, we'll give you a microphone.
It's not a real question, it's an information. I achieved some benchmarking about the question which was raised about the best computing system. And it's my personal benchmark. I can have missed something, but the best is, for my information, the system set up by Peter Tonelato at CBMI at Harvard by using cloud, and it's open source. The informatic treatment, giving the causal cognitive genes, and the last layer of the information, the recommendation for a drug which is activable, matching to the database which are known today, takes 1 hour and takes $35–6.
Okay, it's cheap on top of that. Okay, we take due note. We will share that. Now let's move to the next topic. The next topic, again, as a journalist, is one that is always very good to sell newspapers. It's all about pandemics, influenza. Now we are naturally waiting for the big one. The cycles go up and down, but H1N1, we thought we had something there, but we did not. How can we do personalized medicine to predict and treat such viruses? So I would like to invite Mr. Peter Openshaw, Professor of Experimental Medicine, President of the British Society of Immunologists. He is a specialist of H1N1. He dealt with that in 2009. Dr. Openshaw, you have 7 minutes to explain to us how these viruses can be dealt with in a perspective of the 4 Ps.
Thank you very much. It's great to be here. It's a very different sort of perspective from most conferences, I think, yes, that most of us attend. So I'd like to just tell you a tale of 2 viruses. So this is what I've been doing for the last 5 years, and I would say that it has been the best of times and the worst of times in terms of my experience. The best of times, I think, in terms of the science, the worst of times in terms of trying to get through the regulations and filling out the paperwork. Okay, so the first virus is one of my long-term favorites I've been working on for about 30 years or more, respiratory syncytial virus. So this is a common cold virus that mainly affects infants, and we've recently been infecting adult volunteers. So the reason for doing that is that it's not ethical to infect infants because it causes quite severe disease in a high proportion, but in adults, you and me, it would cause a common cold. So this is showing a study in which we had 61 volunteers at Imperial College, and we gave them all exactly the same virus. So we put the same virus into their noses by nose drops. And 27 of those 61 had no infection at all. We couldn't detect any virus infection. The virus was completely eliminated, and they had no symptoms. Of the 34 that we succeeded in infecting, roughly 1/3 had no symptoms. So 11 out of those 34 had no symptoms. So what's the reason for this intense variation? You know, those who had symptoms, there was a range of severity. So we don't really understand what accounts for that variability. It's not just down to antibody, which I think was the presumption that many started with. So we're analyzing this, this big study using every omic method we can to discover the cause of that variation. So the second virus was the pandemic influenza virus. This is the swine flu virus. It was a unique opportunity for us to study the natural history of a single virus as it spread through a population. A hugely successful virus in terms of its spread. About 75% of people who became infected, according to Andrew Hayward's study in the UK, had no symptoms at all. So only about 1/4 of those who became infected had any symptoms. And of those, there was a huge range. These are 2 patients that we saw at St. Mary's Hospital, where I work in Paddington. One was, this is a caretaker, this is the X-ray of a caretaker rather, who came in having had a relatively short history of flu. He had a high fever, a cough, a bit of an exacerbation of his asthma, but he was treated and he went home within 2 days. On the other side, we have a taxi driver of Pakistani origin who delayed attending medical care. He wasn't really tuned into the medical health system, and he turned up in extremis, very low oxygen saturation, had to be ventilated, was in hospital for 3 weeks. So the same virus, 2 very, very different diseases. So in the face of this, we set up a study supported by the Wellcome Trust and by the Medical Research Council to look at everything we could which might explain this. So we looked at the virus sequence. We know there are some virus strains, high-path strains of influenza, which cause bad disease. We measured viral load. We also looked at the host genome, and we looked at as many environmental factors as we could in order to try to work out what it is when there's a collision between the virus and the host somewhere in the epithelium that causes very severe disease or causes mild disease. We measured bacterial load. We quantified bacteria by 16S. We measured all the mediators, and we looked at the peripheral blood transcriptomics. So in order to try to map what it is that leads from health to severe disease, we thought we needed to study everything, the host, the pathogen, and the co-pathogens. And we ended up with 21 million data items in our database on this rather intensively studied, relatively small number, but a much bigger number, I think, than anyone else has managed to do. One of the things we did especially was a very in-depth inquiry into each medical history, and this took us something like 20 months with 8 workers going over the history independently, trying to validate every detail. Huge study, and a very large number of samples in our biobank. So I can't tell you all the results, it would take too long, but those of you who like to see some data, this is a peripheral blood transcriptomic signature map, which shows that patients in the first 5 days have a rather different signature from those in the later course of disease. So there seems to be a signature change. The cause of that we can understand using this integration of all the data, and we can do network analysis and various other sophisticated analyses. And Charles Offray, who's here in the audience, is, I'm very glad to say, part of the PREPARE consortium, which has gone on from Mosaic, really, to try to validate and understand some of the things that are coming out of this type of study. So there's a huge amount of information to come out of all of this, and I think that from it, I'm sure we'll be able to much more accurately predict who is going to die and who will live, and who indeed will have no symptoms at all when they encounter one of these common viruses. I think the importance of that is that we should be able to target vaccines and antivirals. Just as when I see a patient with chronic obstructive lung disease in the clinic, I give them rescue medication to keep at home. I give them antibiotics and steroids. They know how to take this in need. And in the same way, I think we should be targeting individuals at high risk with, say, influenza. And I think we need much better vaccines. So all of this, I think, is a really good example of being able to personalize, be proactive, and to contribute to participatory medicine. Thank you.
Thank you very much, Professor Openshaw. Thank you. Thank you very much. I think Dr. Buzyn has a question, please.
My question is, do you think that this signature can move with time? I mean, if the patient would have been infected in another period of time with environmental factors, epigenetic factors, that the answer and the signature would have been different? Do you think it could be possible? You mean a different age or a different... At a different period of his life, because he's in a different state.
For sure, for sure. Well, I think it's clear that age is one determinant of how the disease evolves. It's a different disease in children, and we need to stratify for that. There's so many factors that can potentially influence the way in which the signature evolves.
In fact, my question is, you identify high-risk patients with your signature, and do you think that high-risk patient could be high-risk at one period of their life and maybe not such as risk later on because of...
That's a very interesting question. We did publish a paper a couple of years ago in Nature which identified IFIT-M3 gene variants as a major cause of variation in the severity of disease. That obviously won't change through life. But I think that there are also things like the respiratory microbiome, which may change quite significantly. And from the RSV challenge studies, that's coming out as being a really interesting lead that may actually determine whether you get symptoms or not.
Dr. Baudet?
Well, first of all, congratulations. It's a super study and such a nice illustration of pathogen-host interaction. My question is, how specific to the pathogen is the host reaction? And are there elements in the host that would be similar for a variety of pathogens?
I think this is a hunch. I mean, we haven't been able to do the challenge studies in a sufficient number of individuals sequentially to really tell whether susceptibility to RSV equals also susceptibility to flu, for example. We could potentially do that, and we're starting to do some influenza challenge studies now. But I think that's a little way off. I think that in the meantime, my assumption is that the protection is rather personal and individual to different specific pathogens. And I think that's a lucky thing, because if there is a pandemic, we won't all die of it.
Can you also try and model the way that the virus itself mutates as it goes from one individual to another?
For sure. We actually just had a paper in Journal of Virology a few months ago showing that during sequential isolates of the virus, they do behave differently both in cultured respiratory cells from humans, but also in mice. There's a decline in the ability of those viruses to induce interferon, for example, in mouse infection. So the properties of the virus did evolve a bit during the pandemic as it adapted and circulated around the world.
Okay, and a last question. In Quebec, our vaccine for influenza this year was a flop. It didn't work.
Yeah.
You see? Is there a way for your research to try and solve that problem? The fact that it does mutate, from last year to this year, or is this always a chance phenomenon?
That in itself has been a huge ongoing problem for decades, trying to predict where the virus is going to go next. Generally, I think the predictions have not been too bad. This year, it was disastrous, and many of us that were vaccinated actually had flu. But I hope that in the future that prediction may become a bit better. But what we really want are vaccines that protect against all strains of flu and which give longer-lived protection. The vaccines we have, I think, are clearly not ideal.
Okay. Any more questions? Yes, please. Is it possible to get a microphone? Voilà.
So I have a question. If you have data on twins that have been exposed to different environments,
yeah, we would love to be able to do a twin study, very powerful in terms of teasing out the genetic contribution, but this wasn't a study of twins, it was just an opportunistic study. It'd be interesting to do RSV challenge studies on twins, if anyone's willing.
Any more questions? Yes? You can, I don't know, you're far away, but she will give you a microphone. Can you give your name and your organization, please, so that we know who you are?
Yeah, sure. Cormac Sheridan, journalist with BioWorld. I would like to echo the other speakers as well. I think it's a fascinating study. Very curious to know, though, how, say, you can boil down maybe several 100,000 data points per patient into some sort of translatable, measurable biomarker that can be applied at a sort of a system level or in the hospital itself.
Yes, I mean, this is, from the outset, we knew that this was going to be a huge problem, and we wanted to interact with mathematical biologists. I put a lot of effort into trying to get the mathematical biology community to come on board at the start of the study, and it's only now that actually I've managed to find people like Charles who are not only able to do this, but excited about doing it, and we now are moving into being able to analyze our data set using all of these sophisticated techniques, both statistical and other, but that's not really my area, but we're working together on this, and I think there are already things coming out. We've come down to 3 predictive markers which tell us whether a patient will need ventilation, and a different 3 which tell us whether the patients are likely to die. These need to be validated, but I think it is an exciting time.
That's extraordinary. Has that been published?
Yeah.
Wow.
Okay, thank you very much. Thank you very much, Professor Crenshaw, for this fascinating talk. Thank you. Thank you. Now we're going to move on to genetics, genome. I would like to invite Professor Francis Quétier, Professor Emeritus at the University of Évry, France. He's a specialist on genomics. He worked for Genoscope, the National DNA Sequencing Platform, worked on chromosome 14. Now, this is very central, Professor. Genomics, the genes that can tell us, predict if we are susceptible to disease. Can you give us a 7-minute perspective on that? It's all yours. Thank you.
Thank you for your kind invitation. My favorite definition of personalized medicine is as to giving the right drug to the right patient at the right dosage through the right way of delivery at the right time. And in this respect, personalized medicine based on the sequencing of human genomes brings a new and powerful tool to comply with these guidelines. It has now been 9 years since the human genome sequence has been completed, after a decade of research achieved by the international consortium, public consortium, HGP, and have the great pleasure to participate in that saga by sequencing the chromosome 14 within the team of Jean Weissenbach at Genoscope. And the Golden Path, which has been released by GP, cost $3 billion and has been since freely available from the web from the very beginning. And our set of the 22,000 genes constitute our genetic information, which is shared by every individual. A part of this alteration is due to slight difference versions, which are different between each individuals. And this alteration can lead to diseases. During these 9 years, the continuous improvements in both the sequencing techniques and the dramatic increase in the high throughput of robots makes now the sequencing step achievable in 30 minutes, like you heard from Lee Wood yesterday, and for a sum which is a little bit less than $1,000 now. This easy sequencing of individual genomes has opened new tools in the field of medicine and can be divided in 2 main categories. The first one contains 4 points. First, personal genome greatly helps physician and his or her patient when the patient is confronted with a lethal disease, with sometimes and very often close to a fatal outcome. And this is very important because we know now success stories by the score, which have been reported not only for cancers, but for general diseases. In the same medical field, this is a second point, it's now possible to sequence a special category of cells. These are the circulating tumor cells, which occur in the blood. And you can prevent, monitor, survey the possible relapses after chemotherapy or radiotherapy. First, the third point is that personal genomes are very useful because you can predict whether an individual is or isn't likely to develop such or such a disease. And fourth, personal genome can predict for various diseases whether a patient has a good probability to respond to a category of drug, while personal transcriptomes may allow tailoring the dosage and the time of delivery. The second set of application goes in the converse direction from the bench of patients to research, and is useful for fundamental and applied research. Contains also the 4 points. First one is that personal genomes completed by personal transcriptomes gives crucial information about the nature of the gene or the genes. They can be several causing the disease. I would like to underline that out of our 22,000 genes, only half, roughly 10,000, are known today to bring a disease when they bear a mutation. It means that the other half, we don't know, but we highly suspect that they can give rise to some disease, and we don't know which disease match with that alteration for the moment. This identification can use the personal genome. It's rather easily to be done with a rare disease because they are caused each by one gene only. But on the other hand, for most of the current diseases, like obesity, like diabetes, like polyarthritis rheumatoid, and many psychiatric diseases, these diseases are caused by several genes each, and we don't know the number for the moment. So this is here that people and scientists are using large cohorts of the patient genome, try to identify the causative genes, and it's not so easy. Second point is that when a gene, a causal gene has been identified, searchers are going to explain how this gene, alone or in combination with other genes which are interacting with the first one, are responsible for the inception and the maintaining the persistence of the illness. And this is an absolute pre-requirement to imagine a possible therapy. If not, you can't do anything. The third is that personal genomes are also a very good source to identify and develop companion markers, sequence markers, which can be used because they accompany the disease to detect, to monitor, the progression, or better, the regression of a disease. And the last but not least, power is very important. It is the most important. This individual genome sequence can help to stratify the patients because they are heterogeneous. And this is good to have homogeneous cohort for research, as well as to achieve efficient tailoring of the dosage to be recommended for a drug for the regulatory body. Fortunately, both the workflow to apply research knowledge of today to the patient base and the workflow, which is going in the reverse direction to bring clinical information from the patient base to research, are fully identical. They contain exactly the same set of steps in the same order, and they use massively biocomputing and big computing. And moreover, this workflow is generic to all illnesses because it depends on the personal genome, which is unique to any individual. For the moment, the bioinformatic analysis of DNA takes, in the best case, 1 hour of computing, as I told you. When the time is necessary for the DNA extraction from the patient cells, the preparation of the DNA to sequence, the sequence, and the analysis to detect the causative mutation, going back to the physician who made the prescription, it takes now only 3 weeks. This is the target which is assigned to any labs in US. For the moment, the real time is in the order of 2 to 3 months. But this is a target now, 3 weeks. Conversely, the time to go in the converse direction to bring information from the patient's bed to research, translate in new knowledge, and imagine the possible therapy with the preclinical and clinical trials, it's 3 years or more. This is the difference between the two halves of the magic circle.
1 minute left. Unfortunately, time run.
Okay. To finish, the national deployment of sequence-based personalized medicine in France will be the next step. We are involved in launching a pilot platform to test that in various illnesses. And the final step will be to the deployment of sequence-based personalizers in the whole nation. It will involve millions of genome sequencing and analysis, and an expansion of such a scale will require massive investment in robots, in bio-IT specialists, and national partnership with big data, big computing companies. The last message is one sentence. For the moment, as far as I know, the efficiency of the first drug, which is prescribed by a physician to a severe affected patient, the efficiency is less than 50%. We have to do with that.
Mm-hmm. Well, thank you very much, Professor Casey. Thank you very much. Do we have any questions? No? So better, cheaper, faster, I think that's what we have to aim for, right?
Right.
Thank you very much, Professor Casey. Thank you. You can give the microphone to the lady. Thank you. Now, moving from genomics and trying to effectively get better products going on, we're going to go to the products that are right now used. Now, I would like to invite Valérie Raymond-Schwartzman. She is the Companion Diagnostic Program Director at BioVision. She is a pharmacist. Studied at University Claude Bernard Lyon here, so she's back at home. So what can companion diagnostics help us in this 4P paradigm that we're facing? 7 minutes.
Thank you, Michel. So yes, so I'm glad to be with you to present companion diagnostic as a tool to support personalized medicine. And what I will present is really the side of the IVD company, in vitro diagnostic company, and how we are developing companion tests, what are the challenges, what are the benefits, and so on. So if we go back to the definition, and a lot of you define what is personalized medicine, if you go quickly to Google and on Wikipedia, you can see that there is a strong link between personalized medicine and companion diagnostic biomarkers. So really, biomarkers and companion diagnostic are there to support what is the customization of healthcare and the way to stratify patients. So another definition is really coming from FDA, because this is a more advanced notified body to define and give guidelines regarding what is personalized medicine and what is companion diagnostic. So if you have a look to the definition, you can see that companion diagnostic are tests that provides information that is essentials for the safe and effective use of corresponding drug or biological product. So this is really the strong link between the drug and the diagnostic. So therefore, they put here a puzzle. But what I like, it's really more the idea of a marriage or a couple between the drug and the device. So just to give you an idea, and what are your guesses regarding the number of companion diagnostic tests that are registered at the FDA side? Do you have any idea? Because here we talk a lot of personalized medicine, diagnostic tool. We have the feeling that there is so many tools to stratify the patient, especially in oncology. But when it comes to registered tests, so that means FDA approved or CE approved, but at the FDA, this is the highest level of requirements, and this is also the highest level of regulations around that. So do you have a guess? And yes, or somebody in the room? No, so very few, yes. This is. So there is today 20 tests FDA approved, mainly in oncology. So this is what we call BRAF. This is KRAS. This is HR2. There is also some others, but there is also 20. So why? Because from IVD perspective, so we are bringing on the market tests that are easy to use, that could be available everywhere. This is a big difference between homebrew or lab-developed tests. So that means it should be able to test patients everywhere with something that is robust, that is good quality. So this is why the number is so low. And then when I was speaking about this is a couple, the drug plus the test, it's because like a couple, there is good things, there is benefits, and there is things that we need to improve also. So if we go to the benefits of the companion test, this is a huge medical value that this kind of test bring to different stakeholders. So first you have the patient. So here, as we discussed before, this is a way to stratify the patient, to give the right treatment to the right group of patients. I prefer to say group of patients because today we cannot say to one patient, one drug. This is not possible and it will not be the case. Even if personalized medicine, companion diagnostics are really pushed by the new development trend in drugs, because you can see that in 19th there were only 5% of biomarker-targeted drugs, and now this is for 45%. So really an increase. So therefore, we need these biomarkers. So we will stratify a group of patients, and it will also help the patients. You discuss management of the patient, psychological approach, and this is also part of this, because the patient will better understand his pathology, he will better understand his treatment, and then the observance will be better. So this is really a big benefit for the patient. Then also for the physician to prescribe a new biomarker, to prescribe a targeted therapy. This is also an easier decision-making process. This is easy to explain to the patients. Also for healthcare company to have these companion devices, This is a way to decrease the project risk because the duration of the clinical trials will be lower, the cost also, and we will put on the market drug really early. So this is a way to de-risk a project, and some drugs has been rescued by companion diagnostic because at the beginning the clinical trials were really bad and really negative. So Bettina, I see that you are saying yes. So and so it was really a way to rescue this drug because otherwise they would have not been approved. And by understanding better the physiopathology, then we discover some biomarkers that can support the prescription of the drug. And then it ended with a companion diagnostic and a drug, and then we can treat melanoma patients, we can breast cancer. So this, there is also benefits for the IVD companies because then we are putting now the testing, the in vitro testing at the center also of the patient treatment. So it brings value to this test. And also for FDA, for ANSM, for all this notified body, then it's more easy for them to give approval because this is based on more robust data. So for them, we can have a more rapid approval. Also for payers, this is a good way to control the spending, the healthcare spending, because they are more sure that now the right treatment will be given to the right patients with the best efficiency. So this is really an efficient way to manage the cost. So there is a benefit for them. Now, if we go back to my slide, or
you have 1 minute left, unfortunately.
Okay. So then there is here some key issues to manage or some critical factors. So that means the market access and the reimbursement, because worldwide there is not so many platforms like INCA, like in France, and in some countries this is not free of charge. The patients need to pay, so this is out-of-pocket money. So therefore the reimbursement is very important, and this is important of the reimbursement of the drug and of the companion diagnostic at the same time. Also the testing availabilities in order to test all the patients. So therefore, we are developing tests that are commercially available that you can find in every lab, not only in a reference lab like the LDTs, the lab developed tests, or the homebrew tests that are only available in some reference lab because they are not approved. And so this is really the testing availability that is also key. And what we need also, this is this perfect alignment between the launch of the drug and the companion. Also the business model, we have to capture also the created value and to share this between the drug company and the diagnostic company. This is also something that is important. And to come back to the regulatory environment, then also this is a challenge because this is a really high level of requirement. And then I'm done because I see Michel that
yes.
cut. So no, I will not cut, but this is what I would like to highlight regarding a
conclusion then.
Device part.
Thank you very much, Mr. Schwartzman. Thank you very much. Time is of the essence. We only have one speaker left, so I think we'll skip the question period if you want to give enough time to the last speaker. You're very
sure.
nice. Thank you very much.
I'm around, so if
voilà.
you have some question, I'm here.
Okay. Now, time is flying. Thank you very much, BioVision, to give us more time. I think we really needed it. We have so many interesting guests. Now, the last person to talk is Florence Gaudry-Perkins. She is the International Director for Global Government Sector at, we don't know if it's Nokia yet, but it's still it is still Alcatel-Lucent. It's in the news this week. We're going to talk about something very interesting, and I was very impressed when I saw her slides. It's all about mobile health. It is booming, it is very big, and it seems to be very efficient. I think you're going to give us some good examples You have 7 minutes. Good luck.
We're going to have to, try to go fast. So,
Okay.
good morning, everybody. I'm thrilled to be here. Lyon is my hometown, so it's always a pleasure to be back. And BIOVISION is also very dear to my heart. In fact, I want to take 1 second to thank BIOVISION because we won a little prize here 2 years ago via the Catalyzer program, which has turned into a very, very impressive project, I'm happy to report, which I'll talk about in the presentation. I want to do, I always do this when I speak. I'd like to do a quick scan. Can the people who feel they're fairly familiar with mobile health raise their hands? There's a lot of awareness to be raised on mobile health still, and I always kind of like to test it out. So I see no more than probably 10 hands raised in the audience, which is fairly typical, which amazes me because everything that we've talked about since yesterday, mobile health can be a huge tool to accelerate all of those things that you've been talking about. So mobile health is basically the intersection of health and mobile technology. It uses smartphones, tablets, connected sensors, and monitors, as well as fitness trackers and things like that. So this slide here, we won't have time to go through all of these numbers, so I'll just zoom in on 2 or 3, but it sets the scene a little bit. The first thing to realize and to know is that we've reached, in our world today, 7.5 billion mobile connections. This is stupendous. This is more than the world's population. There has never been in the history of mankind a communication technology that has become so pervasive so fast. In Africa, just so you understand, because this is affecting the whole world, it's not just our industrialized countries, 85% mobile penetration is expected this year. Now, what you have to know is that in sub-Saharan Africa, there's about 1% computer penetration. So I don't need to say more for you to understand how incredibly impactful and why this is such a subject of importance. So there's a few key numbers here. Know that as of last year, there were already 100,000 mobile health apps out there. In the US, 86% of people connect via the web to either use health tools of some kind, and 50% of them do it by mobile. So that's something to remember. There's millions of studies out there. Some of them have estimated that mobile health could help save $400 billion by 2017 in industrialized countries, and estimated they could save something like 1 million lives in sub-Saharan Africa by using these tools. So here what I try to do is, it's very difficult, of course, to summarize examples of what's being done in mobile health in one slide because it's affecting all diseases. I chose to give you 2 specific examples on diabetes because it turns out this is what Alcatel-Lucent has focused on and is doing its programs on. So WellDoc is a mobile coaching solution for diabetes, and it's now being reimbursed by FDA, which is very, it's FDA approved and now being reimbursed by insurance, which is very encouraging for the field of mobile health. It's shown tremendous results, as you can see, almost 2% in reduction in A1C values, and reduced hospitalization rates by 58%. Medisafe app is another very good example. It showed plus 81% of adherence on diabetes medication, which is huge when you know what the cost of non-adherence of medication is, estimated in the US at $290 billion a year. So wearables, monitoring devices, I don't think we have time to get into all of that. Huge use in all of these things that you talked about personalized medicine research. Know that it has been estimated that mobile health can help cut clinical trials cost in half and reduce its time also in half. That's beyond all the data generation that we've been talking about. I wish I had more time to tell you about some of the incredible things that are happening in emerging and developing countries in mobile health, but I do want to make this point with this slide here because Alcatel-Lucent has in fact focused on a social innovation partnership approach in mobile health on developing and emerging countries. And here is why. On this chart here to the right, you will see that this is a PwC study. South Africa, India, and Brazil are ahead of the United States and Europe on mobile health. This always catches people by surprise who are working in industrialized countries. And on the left here, you see a chart of a GSMA study where there were more projects in Africa than anywhere else in mobile health. Why is this? I always cite the mobile payment and mobile banking example. Many of you must have heard of this. Many Africans, Africa is way ahead of the entire world on using their mobile phones as a payment method or as a bank account. In Kenya, 85% of the population is using their mobile phone as a bank account or a payment. It's 25% of the Kenya GNP, which goes through mobile payment transactions, if you can believe it. We haven't even begun to see this here. Why is this? Well, Kenya had about 85% rate of unbanked, okay? The reason I cite this example, because it's very powerful, and if you think of how it's going to apply to health, and if you take the lack of infrastructure in Africa, for example, or the doctor-to-patient ratio in Malawi and Tanzania, which is 1 to 50,000, compared to in France, 1 to 300 doctors, here again, you understand why mobile health has a particular significance in terms of what it's going to do to also access to health simply. What we're trying to solve here in our countries has more to do with efficiency and cost effectiveness, but it's also solving problems of access. And now what I'd like to do is just present to you in a couple of minutes. This is this project I was telling you about that actually took partly root here at BIOVISION 2 years ago. It is addressing diabetes using mobile technology. It's happening in Senegal. The field of mobile health has been talked about for over 10 years. It is plagued by the disease that we call pilotitis. There are siloed approaches everywhere. Things are just not going to scale. We believe absolutely that one of the key answers is building multi-stakeholder partnerships with government, international organizations, private sector, NGOs, and citizen sector. The very important partner here is on the right. ITU and WHO signed an agreement 2 years ago, or 3 years ago now, to cooperate actually on helping countries to develop mobile health for NCDs. It's called the Be Healthy, Be Mobile initiative. So they've been a tremendous help to put this project together. The whole government of Senegal is around the table. And you can see the 4 pillars here: M, awareness to help diagnose patients, education, of course, and better management of the disease for patients. The training component is extremely important, and mobile phones can really, really be used for this. There are hundreds of thousands of community health workers in India being trained with mobile phones by simple SMS and IVR for those who are illiterate. So the mobile phone can really be useful for this as well. And I will
2 minutes.
Stay on that. Yeah, and this is just a quick visual, just to give some reality to this is the pilot that we launched last July. It was around Ramadan time, and it was to try to decrease the hospitalization rates and the complications that occur when people fast when they have diabetes. So you see that the messages can be very, very simple and yet have a lot of impact. That was the pilot, and we're moved now. The project is going to be supported for 4 years by the partners, after which we hope it'll run on its own and be financially sustainable and have reached impact. And this is just a little visual of where we stand now. In the middle here, you see the example of a message that's being delivered to health workers for training. We have planned to sort of send about 1 million SMS and quizzes in the next few months and reach 500,000 patients in the first year. Thank you for your attention.
Thank you very much, Mrs. Gaudry-Perkins. Thank you. Thank you very much. We're just on time.
5 minutes?
Is there, any questions? I could take one question because we have about 4 minutes left. No questions? The journalist again, these journalists. Can we give him a microphone, please?
I think actually what you said is very interesting, because I was kind of, the compelling vision that we have around this 4P medicine opens up potential problems around healthcare disparities. Maybe what you're reporting on from Africa shows that that may not be the case. I'm just wondering though how comprehensive an M-Health approach can be, how universal it will be, and how all-embracing it will be in comparison with what the sort of vision that we're seeing for sort of 4P medicines in these very wealthy countries, such as Western Europe, such as North America.
I think you're referring to whether it can be completely comprehensive in the emerging country context. I think what's going to happen here, there are incredible innovations and ideas coming from Africa. I mean, I can cite amazing things, like these 4 young Africans, for example, invented a little device that you connect to the phone where you just stick your finger in it. You don't even have, there's no blood or anything, just through the light connected to the mobile phone. It immediately detects if you have malaria, and that's even before you actually show symptoms of malaria. This is coming from 4 young entrepreneurs in Africa, not from here. So just so you understand, there's, you know, the interesting concept is the frugal innovation that's coming out of these countries as well, because we're trying to solve a cost issue here in our countries. So I can see a lot of things that we're going to be able to apply to our own countries to solve some of our problems. That being said, and this is where a partnership like the one that I described here is interesting, because it's a global and local kind of, you know, sort of partnership. So the innovation is not going to be limited only. As long as you're working on partnership and working both on the global and the local scene, the innovation is going to sort of, you know, come from all places. In the Senegal project, there's a SME out of France which is proposing to build a diabetes database for clinical trials and all sorts of analysis of data. And so we're thinking about bringing them into the Senegal project, just to show you that it's not just only an African project.
Okay. Dr. Baudet has a comment, and we'll close after that.
Well, obviously, this is a fascinating field, and it's great to see it. It's great to see the field evolving. You've mentioned the importance of it and its use for clinical trials, and I'd like to insist on the importance of that. But conversely, I think that we have to be aware also that many of these apps, and particularly in the tracking and diagnostic field, have not been properly tested and have not gone through a proper randomized cluster trial to evaluate the effectiveness, not to mention the cost effectiveness.
Yeah. No, this is a very, very new field, and it's evolving. I heard people talking about walking before running and all of that yesterday, and it's very much true of mHealth as well. There's a lot of, you know, the US is particularly advanced on mobile health regulation, and there's a lot of things that have been happening in the last year because it has to be regulated. It's very, very important. And you're absolutely right. There's a lot of apps out there who are not yet, and, you know, out of the 100,000, there's not so many that are working, especially on the more medical side.
Thank you very much. Thank you. I'll take your microphone. Okay, thank you. So we are coming to an end of this session. I hope that the quality of what we heard and the different views on what has to be done in order to put these 4 Ps into our health system as giving you food for thought. I think that the rest of the day we'll also discuss different issues around the 4 Ps. I would like to thank my distinguished guests. There are 11, I won't name you all, but I thank you very much for your presentation. Dr. Baudet, thank you very much. I think you were pivotal. Dr. Buzyn, I think you saw left. She had to take a train very, very quickly. I thank you both. And on behalf of BIOVISION, thank you very much for inviting me. It's been a pleasure. Enjoy the rest of the day. Thank you very much. Au revoir.